So we're looking at cell membranes, what they're made out of, and how they are able to transport things across them. I don't have a note sheet for this. Instead, I'm just going to ask that what might be helpful would be jotting down the definitions of some terms that'll come through. You'll see those as bolded terms, and it'll be a very specific definition. Those would be good things to know about. So when we talk about a cell membrane, every cell, whether it's bacteria, plant, animal, fungus, anything, has a cell membrane, and that's what separates the outside from the inside. And all cell membranes are made from a very, very large number of these smaller pieces called phospholipids. And a phospholipid, as the name implies, is a lipid. It's a polymer of lipids, and it's made from the monomers as this phosphate group and two fatty acid tails. So we had previously seen fatty acids as one of the main types of monomer of lipids. And what's notable about those fatty acid tails is they're very hydrophobic. They're non-polar, so they do not mix with water. Meanwhile, the phosphate group here is hydrophilic, so it can interact with water. It doesn't. It's not repelled or pushed away from a polar substance like water. So these phospholipids, like I said, are the building block of the cell membrane. It turns out that when you put a bunch of them next to each other, the hydrophilic polar heads tend to stick together and the non-polar tails tend to stick together. And that's just because the like with like, that the non-polar ones will tend to congregate near each other and the polar ones will as well. However, if we were to take this and stick it into a bucket of water, you're not going to find those hydrophobic tails wanting to interact with that water. And so we actually don't see the cell membrane generally as a single layer like this, but rather as a double layer. And here you can see how there's an outside layer and an inside layer. This would be wrapped all the way around the cell. And on the next slide, you'll see a picture of that. And what that does is it creates this middle area to the cell membrane that's very non-polar. It does not mix with water, and therefore is able to sort of keep the outside on the outside and the inside on the inside. So this is called a phospholipid bilayer. It's two layers of phospholipids with the hydrophobic tails in the middle, forming a non-polar region that separates the outside from the inside. So here we can see this expanded to be an entire cell. Remember, a cell is a three-dimensional thing, so this is just a flat version of it. It would actually go sort of like into and out of the screen as well. This is a pretty profound thing, though, because what happened is in the beginning of cellular life on the planet, it was the first time that we created an inside. And that's kind of a weird thought to think about, but the point of it is that prior to that, you have an ocean of stuff, and there's all sorts of biochemicals in there, and maybe little amino acids and components of proteins and things like that. But you don't really have any separated areas. And as we're going to continue to learn, our cells are driven by chemistry, and chemistry requires change. And the best way to establish a change or to be able to control a chemical reaction is by being able to separate these things across some sort of barrier. Because if you have a difference across a barrier, then you can sort of control what happens. You can control what moves in, what moves out, and you can drive a process. And so the earliest cells being these phospholipid bilayers, it's the first time we have an inside. Before, it was just one big ocean. Now it's one big ocean and the inside of this cell, and the inside of every cell, in fact. So we see our phospholipid bilayer, and we can say it's kind of a watery environment on the outside. It's a watery environment on the inside, but they tend to stay where they are, those environments, because the water can't cross this non-polar middle. So everything on the inside stays in, everything on the outside stays out. Now that obviously kind of poses a problem, because if nothing can move in or out, then what's the point of it? And so here we see a situation where we have a whole bunch of these little red dots on the outside, and maybe that's like a small molecule like oxygen. Oxygen's super tiny. Oxygen gas, I'm talking about O2. It's super tiny. It's non-polar, and therefore it actually can pass right through the cell membrane, because remember, it's that non-polar barrier in the middle that normally keeps stuff out. Something like oxygen can simply diffuse into the cell. We'll pass right through that membrane. So this is the example of just plain old diffusion. And diffusion is the movement of molecules from high to low concentration. So an area where you have like a lot of something and it tends to just spread out. It does not require any energy, and in this case it didn't need any help to get into the cell. So this is what we call simple diffusion, or more often we just say the word diffusion. But what if we had a circumstance where maybe we have something that's a little bit bigger, or maybe it's polar and therefore can't pass through the non-polar inside of the membrane? In these cases, you're going to need a little bit of help in order to get things into the cell. And in this case, what's happening is we have a protein. And so here's a protein that I've added to the top of this cell. And what this protein does is it basically forms a channel. In fact, we call them channel proteins. It forms a channel for this particular thing to come into the cell. Maybe this is some sort of salt molecule or a glucose or something. It's something we need inside of the cell, but it can't pass through the cell membrane. In this case, we have a protein which is helping to facilitate moving that into the cell. Now, we still give this to the title diffusion because, again, facilitated diffusion is the movement of molecules from high concentration, an area where you have lots of it, spreading out to an area where you have less of it. The important difference here is that it has the help of a membrane protein. So facilitated, means like it's helped. So in this case, it's helped by this protein. It does not require energy to do this. This protein is basically just forming the right shape, like a hole in the side of the cell membrane that things can pass through. You can think of it as a doorway into a cell. If a cell is a room, here's the door, and that door fits certain things. It doesn't fit all things, right? You could imagine trying to move a couch from one room to another and running into the issue where the door is not wide enough or something like that. So similarly, in facilitated diffusion, that protein is really looking for a particular thing that fits just right through that channel. Another circumstance is one where maybe we don't want to move things from high to low concentration. Right? Because maybe we need like a lot of something inside of this cell. This cell really needs a lot of pink squares inside of it in order to survive. In fact, if it doesn't get enough pink squares, then it simply won't survive. So in this case, we have a different process, and it's one that's going to require a little bit of energy. And this process is called active transport. And here you can see a little ATP molecule. If you remember, that's the adenosine triphosphate is the chemical sort of energy of the cell. And in this case, if we jump between these two pictures, you see that we went from having kind of even amounts of this pink square spread throughout the cell and spread inside the cell, outside of the cell. But then we turned on that active transport and it moved more inside the cell. It went against the concentration gradient, which requires energy to happen. Now, it's worth it to the cell because if the cell needs pink squares in order to survive, then that's just something it's going to have to use its energy to do. And we do this inside of our cells all the time in order to create the right amount of certain substances inside of the cell as part of homeostasis, so we can maintain the needs of the living organism, but that comes at a cost. So active transport, like the name implies, requires energy, and it is the movement of molecules from low to high concentration. So you're taking something that's totally spread out and you're bringing it together into one place. And of course, that's going to require energy. So these were just a few things about the cell membrane, the structure of the phospholipid and how it arranges itself into this bilayer. And then we were talking about diffusion, which was simply movement of molecules from high to low concentration. It can go right through the membrane, doesn't require any energy. Facilitated diffusion, which was the case where we saw the movement from high to low concentration, but with a little bit of help from a protein. And then the last one, which was active transport, where here we have a protein that might be pumping things into the cell, and that is typically that will always be from low to high concentration, and therefore it requires energy. So these were the 3 different types of cell transport that we're talking about today, and that's it. If you have questions, please email. There is an exit slip to complete. Thanks.